Good morning. It's Friday. It's 8:30, and what we do is to really every week celebrating vision research, and I'm just really excited to see all of you. And today we have a phenomenal scientist, but also a contributor to our research. Patsy was very generous with her work, and I'm sure some of you already using mice characterized by Patsy, and on top of her really outstanding research. So we're truly delighted to have her today, and I think Hernan is the one who is going to introduce.
Hello and good morning. My name is Hernan Gomez, and I am a second-year graduate student in Dr. Krzysztof Palczewski's lab here at the Center for Translational Vision Research at the University of California, Irvine. It is my pleasure to introduce today's seminar speaker, Dr. Patsy Nishina. Dr. Nishina completed her MS and Ph.D. in physiological biochemistry at the University of California, Davis. Following this, she was a postdoctoral fellow and assistant research biochemist at the UCSF Benioff Children's Hospital in Oakland, California. After, she worked as a research assistant at the Jackson Laboratory in Bar Harbor, Maine, ascending to become a full professor of the Jackson Laboratory. In addition, she is also professor at Tufts University School of Medicine of the Tufts School of Graduate Biomedical Sciences in Boston. With over 120 peer-reviewed publications and over 15 book chapters and reviews, Dr. Patsy has had many academic honors, including being speaker of the Distinguished Lecturer Series at Columbia University and the keynote lecturer of the first International Symposium on Usher syndrome and related disorders. In addition to her academic honors, she is a member of the Association for Research in Vision and Ophthalmology and member of the COMP2 steering committee. Her current work as a principal investigator involves projects looking at genetic modifiers of retinal disease, translational vision research models for subretinal fibrosis, and looking at mouse models bearing human AMD GWA alleles. Now, please join me in welcoming Dr. Patsy Nishina for her talk, Mouse Models for Dissecting Genetic Interactions and Complex Retinal Diseases.
Thank you very much for that introduction, and I'd like to really thank you all for the opportunity to share with you some of the things that we've been doing in our laboratory. My talk is entitled Mouse Models for Dissecting Genetic Interactions in Complex Retinal Diseases. And the complex retinal diseases that I'll be discussing are those associated with age-related macular degeneration and macular dystrophy. So we take a genetics approach in our laboratory toward understanding gene-gene interactions and common pathways that are induced by primary mutations that lead to disease phenotypes. At the heart of our work are mutants, and these mutants can come from a variety of sources, chemically induced, spontaneous mutants, as well as those that are genetically engineered. Once we have a mutant in hand, we do careful phenotypic characterization, looking both at disease progression as well as pathogenesis, focusing on morphological changes as well as biochemical changes. And then we do functional genomic studies and genetic modifier screens to identify those pathways and molecules that interact with the primary mutation to affect disease phenotype. So for the purposes of this talk, I'm going to be focusing on two aspects of our work. One is transgenic mice, where we've engineered human AMD GWAS alleles into these animals. And then I'd like to finish up with a description of some of the chemical-induced models through the Translational Vision Research Models program that is cited in my laboratory. So I'm sure all of you know this all better than I do, but I always like to start out with this type of slide just to describe the fact that AMD is a leading cause of elderly blindness. And you can see in this left-handed panel the central vision of this individual that has been diagnosed with AMD is obscured. So you can imagine that the quality of life of these individuals is significantly impacted. And so I think anything that we can do toward understanding the pathogenesis and the mechanisms through which it's functioning would be very important. We know that 50 to 70% of the total variability in disease risk is attributable to genetic factors, and 30 to 50 loci have been identified that are associated with AMD risk. And this disease is likely to be multifactorial. That is, that either different variants lead to disease phenotype, or an interaction between disease variants lead to phenotypes. There are two forms of AMD, wet and dry. 95% of individuals that are diagnosed with AMD have the dry form. And this is a slowly progressive late onset disease. And it is this feature of the disease that makes it much more difficult to develop therapeutic approaches, because it's difficult to deter or to assess efficacy. So our hope is to understand the mechanism of pathogenesis, to identify early biomarkers of this disease, to use as surrogates to have a platform in which we can have more effective therapeutic targets. So I'm sure all of you are aware that mice don't have a macula. I've been reminded this numerous times in the grants that I've applied for. Mice don't have macula, so how can you use this as a model? Well, it's probably about 95% of all retinal diseases that have been identified in humans, when transferred into mouse, also entire recapitulation of the disease, we often see subphenotypes that can be interrogated, and understanding and studying these subphenotypes may give us ideas about the mechanisms through which they occur. The other thing that we can do with mouse, and other people have done this to great effect, is to take the human variants that have been identified in humans for AMD and place them individually into a fixed genetic background to examine the effects of the disease. And this is really important because when you think about the human population, there's so much going on with genetic heterogeneity that is there, that being able to isolate those variants, I think, is very important. So there are two loci that have been identified that is responsible for approximately 50% of the disease risk in AMD, CFH, and a region on chromosome 10q26. This region contains two genes that are in disequilibrium, ARMS2 and HTRA1. And this locus actually carries the highest risk for late AMD at a relatively early age. And Dr. Den Hollander and her consortium have shown that this risk variant, or risk variance at this locus, acts as a strong catalyst for disease progression. The problem is ARMS2 and HTRA1 is in such strong linkage disequilibrium that it's been difficult to know whether or not it's ARMS2 or HTRA1 that is causing the increased risk, or whether or not it's a combination of both. The other problem is that ARMS2 is a primate-specific gene. So there aren't very many models that have expression, or there aren't any models that have expression of ARMS2. And so it's difficult to be able to model the disease that it might affect. So we decided to hunker down next to this chromosome 10q26 region, because we felt that this is an area in which we can use the mouse model to, and the powerful tools associated with it to understand what ARMS2 might be doing. So there are two groups that have made transgenic ARMS2 animals, taking the human gene and placing them into mice. Both of them used promoters, ubiquitous promoters. One inserted it into the Rosa locus and the other randomly inserted into the genome, mouse genome. Neither of these models were reported to have a retinal phenotype. So when thinking about what we should do for ARMS2, we selected the ARMS2 A69 variant, which is thought to be the allele that is conferring susceptibility, and we inserted it upstream of the HTRA1 mouse gene into a region that was found to be homologous between human and mouse. Then we included as much of the 5' regulatory region as possible when we were developing our construct, and we were very happy to see that ARMS2 is expressed in eye, placenta, and brain in mouse as it is in humans. So what are the phenotypes? We take a number of methods to assess disease. First, fundus examination. And here you can see, I'd like to focus you on this panel here. You have animals that are 9 months old or 18 months old that carry the ARMS2 A69 variant. And you can see that there appears to be a progression of this disease phenotype. We think that these spots represent immunological cells that have been activated and migrated to the subretinal space. Here we see IBA positive cells that are sitting on top of the RPE monolayer. So it appears that the allele causes progression of disease with time. So one of the concerns that we had was, how do we know that what we're observing is specific to this variant allele versus just producing a non-native protein in mouse? And so we went back and did gene editing of the ARMS2 A69S to form the wild type ARMS2 A69 animals, and then looked in their eyes. And here, this is at 9 months of age, and you can see that it doesn't have the spotting phenotype. However, if you shift over to the black 6 animals that are 18 months of age, these animals show a spotting phenotype, albeit not as severe as in the A69S animals. This suggests to us that ARMS2 A69S is making a normal aging process more severe in the animal. If you look at these animals histologically, we see duplication or hyperproliferation of the RPE, as well as subretinal cells that are nucleated in the subretinal space. And we see areas of debris collected in the ARMS A69S animals. And by TEM, in the subretinal space, we see these fields of debris associated with nucleated cells, and we also see lipid droplets. So we think that, or our hypothesis is that perhaps ARMS2 is doing something to the integrity of the RPE cells. And so we wanted to know what types of changes are occurring in RPE. So Gail Collin in our laboratory adapted a method that was that isolated or separated the posterior retina. So using hyaluronidase and vanadyl ribonucleoside complex to inhibit or degeneration of RNA, we were able to separate out retina from RPE and choroid sclera. And we wanted to know whether or not these fractions showed significant contamination or not. So we did RT-PCR with retina-specific gene, RPE-specific gene, and then gene that is expressed in the choroid. And you can see clearly here that these are enriched fractions. So the retinal fractions express arrestin 3, RPE fraction, GR, and the choroid fraction, collagen A1. So we're pretty confident that this was a good separation of the eye cup. And so we went on to do global transcriptomic analysis. And this is our data specifically for RPE. So this represents RPE that was isolated at P14. And interestingly, we see upregulation, if you will, that is significant in 2 different types of pathways, cell-cell junction genes, as well as cytoskeletal and intermediate filament organization genes. We now want to understand how it is, or whether or not this is a response to the allele, or whether or not this contributes to the disease phenotype that is observed. And that is ongoing work in our laboratory. We also noted that there was a disruption of lipid profile in these animals. So here you have BLAC6 and ARMS2 A69S at 12 months of age. And you can see positive staining by Philippin in the RPE region in these animals. We took our first foray into spatial metabolomics, specifically looking at lipids in these animals. And we see that there's a difference between BLAC6 and ARMS2 A69 as animals see increases of phosphoinositide, ceramide, and sphingomyelin. We don't know what this signature change is, what its significance is, but that's something also that we need to look at in the future. What's really interesting is that when we looked at the transcriptomic data, the P14 data for RPE, what we noted was that there were some mitochondrial genes as part of the electron transport chain that were significantly downregulated in these animals. And so you can think if you disrupt energy metabolism within the eye, that might lead to a disruption of lipid profiles. And when we looked at the retina RNA from these animals, we saw that mitochondrial biogenesis, organization, and lipid oxidation genes were significantly downregulated in ARMS A69S relative to BLAC6 controls at P14. Together, this would suggest that something is amiss, that ARMS2 is causing some effect in the retina of these animals, and perhaps these are going to be early biomarkers of the disease. But we wanted to be certain, again, that this was not simply due to the fact that ARMS2 was being expressed in the retina of these animals. So Naveed collected RPE samples from BLAC6, ARMS2 wild type, and ARMS2 A69S animals. And you can see in the 3 of the 4 genes that we tested that there was a significant downregulation of ND4, ND2, and cytochrome B in the ARMS2 A69S. And in the case of ND2, it was also significantly different than ARMS2. So this is not something that was occurring just because we overexpressed or were expressing the transgene ARMS2 in the back of the eyes of these animals. But it may be due specifically to this allele. So just because you have a transcriptomic change doesn't mean that that translates to a functional change. And so Naveed collected posterior eye cups, again, from these same mutants and wild type, and looked at NAD levels as well as NAD and NADH levels. And you can see here that, again, ARMS2 shows a downregulation of total NAD levels as well as NAD-NADH ratio in these animals, suggesting that there might be a mitochondrial defect that results from the transcriptomic data that we showed you. Of course, we have to do a lot more work to confirm our results. And that's the what is it called? the goal of this project, to figure out what it is, what are the underlying mechanisms that are being induced in the case of ARMS 2. So we have a lot of other questions. Now that we have a model in hand, one of the things that comes to mind is how it is that ARMS2 A69S is acting. So in the literature, there's a lot of conflicts. Some people say that it's acting through HTRA1, that you see an elevation in HTRA1 in individuals that have this variant in donor samples. And then others see no effect at all. And so we looked in eye cups, right, in a mixed tissue at HTRA1 levels in this variant. And it turns out that there is no effect on expression. However, if you isolate the RPE specifically, we see an elevation of HTRA1. Of course, this doesn't mean that ARMS2 is acting through HTRA1, only that it has an effect on HTRA1 levels. So what we're gearing up to do is to make a tissue-specific or RPE-specific knockdown or hypomorphic allele of HTRA1 in the presence of ARMS2 A69S, and asking whether or not there are any effects on the transcriptomic data or the end-stage disease phenotypes that we're observing. We also want to know what the early subphenotypes are in this animal. And one other aspect that I'm interested in, because I'm interested in gene-gene interactions, is that in the literature, it's been shown that ARMS2 A69S sometimes shows an interaction with CFH, which might potentiate disease. And in other situations, it's been suggested that there is no effect. So we generated the two CFH models that carry mutations that are segregating in large AMD families that are segregating for AMD. And we made the mouse correlate of these variants. And what's really interesting is that if you look at C3 levels in these CFH models, you can see that the D108G allele looks really similar to BLAC6, but the CFH animal that is carrying the R71C, you see very low levels or no C3 in these animals. So the question goes back to, is it that you have to have a particular allele of C3H to interact with ARMS2 A69S to potentiate or increase risk of disease? And so we can do this now that we have these animal models. And finally, there are a number of clinical trials that are ongoing that have been used, that are being used in dry AMD. And one of these are drugs that are thought to enhance mitochondrial function. And so we're hoping to be able to use that mouse in our model to see whether or not we can attenuate or ameliorate the changes in the mitochondria and end-stage disease phenotypes in these animals. So we have a lot to do, and if anybody has any suggestions of what else we might do or would like to collaborate, we'd be more than happy to do that. I'd like to shift gears now and talk about mouse models to examine subphenotypes of macular diseases. And I'd like to talk about two models that we identified through our ENU mutagenesis program. So the first model is TVRM5, Translational Vision Research Model 5. And it was detected by fundus examination as having these dense lesions, primarily in the dorsal region. We identified it as a mutation in CTNNA1 that changes a leucine to proline, so a linear structure amino acid to a ring structure, suggesting or indicating, I guess suggesting, that perhaps it would affect the function of CTNNA1. Together with Anneke den Hollander, we were able to show that mutations in the human population in CTNNA1 leads to macular or is associated with butterfly-shaped macular dystrophy. And subsequent to our report, a number of groups have shown similar results. So ctNNA1 stains in the cytoplasm as well as in the cell junctions of RPE cells. And as such, functions in cell adhesion and also as a molecular sensor organizing intra and intercellular signaling. And affects then cell migration, proliferation, and differentiation. So it's not surprising that it has such a basic function, because it has such a basic function, that when you make ctNNA1 null in the mice, they're embryonic lethal. In our case, we were dealing with a hypomorphic allele in which we have a point mutation. So those animals are viable through embryogenesis, and we then can observe the adult phenotype. And in this case, it was in the eye, and we see these lesions in the retina of these animals. So like ARMS 2, interestingly, we see a phenotype of duplication of or hyperproliferation of pigmented cells. And we also see nucleated, pigmented, and non-pigmented cells in the subretinal space. And we also see these lesions that appear to be encapsulated within the RPE. And so if you cut through, do serial sections, we note that in the area prior to these lesions, you can see that there's hyperproliferation of the RPE. And then you see this lesion. And then you go out of the lesion, and then you can see areas of hyperproliferation. So Mark noted this when he was looking at these animals, that when you orient the eyes and look at the location of these lesions, and here he superimposed different animals, lesions that were found in different animals, identified by different color markers here, that they all seem to localize centrally to the dorsal region of these animals. So it's been previously shown that in pigs and dogs, there is what is called an area centralis that is identified by increased neuron density. And Dr. Aguiar has shown in dogs that a gene known to cause macular dystrophy in humans, when mutant in dogs, also causes a topographical effect in which he sees lesions in the area centralis of the dogs. So mice have previously been shown to have increased ganglion cells and cone photoreceptors in this dorsal region. So we hypothesize that the TBRM5 lesions are identifying the area centralis in mouse. And this becomes important because as we examine mouse models with AMD variants or what have you, we should try to orient the eyes so we can look particularly in the region to see if there are early phenotypes that couldn't be detected or pathologies that can be detected in these eyes. When we look at the lesions by staining methods and autofluorescence, you can see that these TBRM5 lesions autofluoresce, and they stain for alizarin red, which is stained for calcium, as well as for philippin, which stains for unesterified cholesterol. So these lesions, despite the fact that they aren't localized to where we would think of as drusen, they share some characteristics of these lesions share some characteristics. What we really need to do is to isolate these lesions and do a compositional analysis to see how similar or different they are. So the other point to be made is this. ctNNA is a gene that is expressed in epithelial cells, in neuronal cells, and in testes. So when we examined the TBRM5 mutant, the only overt phenotype that we observe is in the eye. And the question is, what is it about the eye that makes it permissive for the formation of these lesions? Is there something in the genetic milieu of the RPE that is interacting with TVRM5 to cause the disease phenotypes that we observe? So we decided to take the tact of looking for genetic modifiers, because genetic modifiers is something that we can do very handily in mice that is difficult to do in humans. And these identification of such modifiers can provide insights into the function of molecules, as well as insights into the pathological changes during disease. So the idea here is that you take your primary mutation, in our case, TVRM5, and you put them in different genetic contexts. That is to say, we cross them to different inbred strains. And then we look for a difference in phenotype. If a difference in phenotype is observed, we conclude that there must be something that is in that genetic background that is interacting with the primary mutation. And we can use the crosses in the mouse model to identify the molecular basis of those modifiers. So what we did initially was to take the TVRM5 mutant and cross them to 5 inbred strains. And these happen to be 5 of the 8 parental strains that make up the collaborative cross and diversity outbred panel. And you can see clearly that there's differences among these animals, and that 129 shows this interesting spotting phenotype, and in NOD, we see this really fantastic phenotype, suggesting that both 129 and NOD have something in their genetic background that is interacting with TVRM5 to lead to these disease phenotypes. So Gail and Lisa decided to take the TVRM5 mice and cross them onto the NOD background and look longitudinally to see what the progression of the disease was. And this fundus phenotype is not there at 1 month of age. However, by 3 months of age, you see significant modeling in the dorsal region of these animals, suggesting that this is a progressive disease. And Gail wanted to be absolutely certain that this was not due simply to an interaction of BLAC6 and NOD. So she looked at the F1s between BLAC6 and NOD, and we didn't see any lesions in these animals. However, in a back cross, what she found was that there were 2 populations of animals, those that had moderate lesion formation and animals that had really significant disease. So she took that back cross DNA and did a mapping cross, she and Kyle, and they were able to determine that there was something on chromosome 6 that was regulating this phenotype and interacting with TVRM5 to cause this modification of this phenotype. For those of you that have done mapping previously, you'll know that in these kinds of mapping crosses, the areas that are identified are huge, right? So you can have like 300 to 500 genes within the critical region. And so therefore it's really difficult, well, it's not difficult, it's nigh impossible to try to figure out exactly which of these genes are the cause unless you do very, very large crosses. And so what we decided to do was to use a resource, diversity outbred mice that are predicated on 8 inbred strains that had been intercrossed to form the collaborative cross strains. These collaborative cross strains were then outcrossed to produce the diversity outbred mice. And these mice approximate the genetic heterogeneity that is observed in the human population, and they're likely to be heterozygous at every locus across the genome. Because of this genetic heterogeneity, these mice identify very narrow regions for genes of interest when they're used as a mapping resource, sometimes down to 4 or 5 genes, and for those that are very lucky, down to a single gene. So our plan is to take TBRM5 mice and cross them to 2 to 400 DO mice. And from these crosses, we'll select one offspring from each cross and then genotype them genome-wide, and then do as many phenotypes or assess as many phenotypes, quantitative phenotypes as we can in the 2 eyes that are available to us. So we'll do ERG, OCT, histology, immunohistochemistry, and flat mounts to look at RPE morphology. And if anybody has suggestions as to other phenotypes that we might look at, we really appreciate the input. But with this size of a cross, we expect that we'll be able to detect 80% of the modifiers with effect sizes of at least 1/2 standard deviation. So we're very, very excited about doing this experiment. And just to show you some of the pathological changes that we might be able to use, we see these lesions, abandoned lesions in the dorsal region, and we're hoping that we will be able to quantify this using AI technology. We also can measure the morphological changes in the RPE, as well as the nucleation status and the localization of where these aberrant cells occur. We can also do histological staining. Here you can see that there's accumulation of lipofuscin material, but perhaps we'll be able to measure the area of these debris fields as a quantitative measure. But whatever the case, I think that this experiment will provide a lot of information about what it is that TBRM5 is interacting with, potentially in the RPE and in the retina in general. So as we obtain these map positions, one of the things that we can do is to compare them to the global transcriptomic analysis data that we have for these animals. So like the ARMS mice, we tested at P14, 5 males and 5 females that carried the mutation in CTNNA1 and compared them to BLAC6. And what we found were the top functional pathways that were differentially expressed were intermediate filament cytoskeleton organization processes, pathways, as well as molecules associated with extracellular matrix and extracellular structure organization. So as we identify these regions that carry genes of interest that are modifying the disease phenotypes, we could look at this list that we assembled through global transcriptomics as potential candidates. So I'd like to end with the second model that we've identified through ENU mutagenesis. And what's very interesting about this model is that it shared amazing similarity to that of TBRM5. So these animals have the discrete lesions in the dorsal area here in TBRM5 and in 283. And these represent, we also see, for example, here, the lesions that are pseudo-colored here. And if one then magnifies and goes in, you can see that there appears to be pileup of phalloidin positive staining. I'm suggesting that there might be active proliferation in these areas. We also see hyperproliferation of RPE cells, and in both we see these lesions that are encapsulated within the RPE. Looking at RPE defects for both TBRM5 and TBRM283, we note that there are morphologically affected cells, and there are cells that are multinucleated in both TBRM5 and 283. Another shared feature of these two models, is that they have characteristics of subretinal fibrosis, a phenotype that is often observed in age-related macular degeneration. we see positive serous staining for fibrotic tissue, as well as positive staining for collagen 1 and smooth muscle actin in these lesioned areas in both TBRM5 and 283. And as I've shown previously, TBRM5 mice have positive staining for cholesterol accumulation in the RPE region, and that is also true of TBRM283. And when you look by TEM, we see these areas of debris in the RPE and a reduced basal infolding in both TBRM5 and in 283. So they share a lot of phenotypic similarities. So we've identified subsequently that this gene is due to a mutation in LRAT-D2, which is lecithin retinal acetyltransferase domain containing 2 protein or gene. The protein localizes to the plasma membrane and at cell-cell contacts in the cytosol, much as we've observed for ctNNA1. And what's interesting is that somebody did a blind screen using yeast-2 hybrid analysis with ctNNA as bait, and they were able to pull down LRAT-D2. So there appears to be a direct interaction between ctNNA and LRAT-D2 that led these investigators to suggest that LRAT-D2 was a binding protein for ctNNA1. And the very coolest thing about this gene is that it is localized quite close to a suggestive GWA hit for early stage AMD. So the question is there a genetic interaction between ctNNA1 and LRAT-D2? And so we did candidate gene testing where we crossed the TBRM5 animals to make a compound het for TBRM283 and TBRM5. So in animals that carry 1 copy of TBRM5, you can see that there are these lesions in one eye here, but not in the other. There might be one here. But when you look at animals that are compound heterozygous, you see more of these lesions that accumulate in the dorsal region, suggesting then that interaction between ctNNA1 and LRAT-D2. So perhaps these 2 mutations are defining a pathway that may be important in the formation of these lesions in the dorsal region. So what have we learned? I'm hoping that I've been able to convince you that mouse models can be used to examine specific subphenotypes associated with macular diseases, including mice bearing AMD variants. And that these models allow us to examine the pathological effects of these mutations as well as disease progressions, and hopefully will allow us to identify early biomarkers of these disease phenotypes. And interestingly, there are common pathological features and transcriptomic profiles that appear to be shared among these different model systems that have RPE defects. And it is our hope that with use of the unique models that are available in mice, that we'll be able to identify and elucidate the pathological pathways and mechanisms that are induced by genetic mutations and variations in genes, for example, the GWAS alleles. So with that, wow, I really speeded up and talked fast. However, that would leave a lot of time for discussion, and you folks can share with me what it is, your ideas of what we could do with these models. So I'd like to end with thanking our collaborators. Greg Carter is a phenomenal computational scientist that has been working with us for all of our analysis. Anneke den Hollander and Robin Robin we worked with them for the ctNNA1 story. Neil Peachy is our go-to for anything to do with ERGs, and he's really a wonderful collaborator. For those of you that have worked with him, you'll know that. Martin Perra and I have begun studies. We're hoping that we will be able to take the different ARMS2 alleles and variants in the region and look at their effects in culture from human differentiated iPS cells into RPE cells and look at the effects in cell culture. And Marius Ufing has agreed to throw in with us to look at the protein aspects of ARMS2, and in particular the ARMS2 A69 allele. And I'd like to point out that without the fantastic support from NEI and foundations, we wouldn't be able to do the work that we do. And this is a list of our current members. Bo Chang and Jay Ping run the iMutant resource. And I'm really sad to say that the EMR, the iMutant resource, is closing its doors in July as Bo Chang retires and moves on to greener pastures. I'd like to acknowledge Gail Collins and Lisa Stone, the cornerstone of our laboratory. Gail Collins is lab manager, Lisa Stone is our colony manager. Without them, we wouldn't be able to do the work that we do. And Vicki Kennedy has joined our group to help them. And I'd like to acknowledge Mark Krebs, who has brought to us tremendous depth in imaging, as well as thoughtful discussions on what it is that these molecules might be doing in the eye. Navdeep Konya is a postdoctoral fellow, which she's been very pleasant and taken on really a lot of projects. And she would be so happy if somebody came to visit and work with us. And finally, I'd like to thank Kyle and Anna, two student interns that have contributed to various aspects of the projects throughout the years. And finally, the Jack Scientific Services, which we are absolutely indebted to in doing the work that we do. And so this is a picture of our group on a summer's day, and you can see Frenchman's Bay behind. So any of you that have come to Maine, you know that it really is beautiful, both in the summer as well as in the winter. So with that, I'd like to end, and if anybody has questions, and later on, if anybody has comments, I would appreciate receiving them via email.
This was such a high quality work and presentation. Thank you very much. I have a very quick question about this LRRD2. Do you know if it has any enzymatic activity, this protein?
I think people have looked and they say that it does not have enzymatic activity.
Okay, so we have at least 10 questions from the room, and I see that our brother Anand Swarup is open microphone, so I'm sure he has some thoughts.
Hello, how are you? I haven't seen you such a long time.
Hi, Patsy. How are you? Good. Very nice talk.
Okay, let's go to Caroline.
Hi, Dr. Nishina. Thank you so much for your talk. My question was regarding developing mouse models with human sequences on it.
Sorry, I didn't hear your question.
Okay. Hi, Dr. Nishina. My name is Caroline. My question is regarding developing mouse models with human sequences on it. We see a lot of regional homology, but still have like some amino acid changes. Were you able to perceive any disturbances in protein structure, or like for genes that have different expressions in different tissues, like more broadly, like animal homeostasis changes with these interactions?
You know, I think this is actually an area that Dr. Bochowski is probably more able to answer than I could, because we're not structural protein people at all. Basically, what we do is genetic studies, and from my perspective, we see these phenotypic changes, and it would be fantastic, if the world was such that we could take all of the experts in different areas and bring them together to face on a single question. I think this, in this case, yeah, we have not looked, but I think that that's definitely something that someone with expertise should look into, as to whether or not structural abnormalities might lead to a difference in activity or function within tissue. That would be really a good thing to do.
Ready.
Hello, so I'm interested in the TVRM5 mutant you presented, how on the one genetic background, within 3 months, there was the degenerative phenotype. Did you look at the progression in on other backgrounds, whether that was just a slower time course or whether it never developed at all? And how might that be affected by light exposure?
Okay, so we have not done light exposure studies in these animals. And we have with other strains, for example, like mutations in rhodopsin, where we expose to light, and some develop very rapid degeneration and others not so much. And your question of looking at different genetic backgrounds, I think that's the whole point, right? Most people look at mutations on the Black 6 background or a mixed background, right? I think it's really, really important to try to place these mutations in different genetic contexts and ask whether or not there are differences among these animals. And if you can identify these differences, then you have the possibility of actually identifying the molecular basis of what is causing those differences. So we saw, I showed you that we tested against 5 different strains. The number of inbred strains are limitless, but you can choose, for example, when you look at the family trees of the different mice, you could potentially choose different branches of mice and different strains to be able to try to maximize your effort in looking for what these modifiers could be. With the exception of 129 and NOD in our cohort, we didn't see anything insofar as fundus examination. Unfortunately, we didn't actually look at the retina of these animals, morphologically, what was happening in the retina of these animals. And that's something that we're going to be doing both in the NOD background as well as some of the other backgrounds that we are testing currently. Yeah.
Geralda?
Hello, it was wonderful, really wonderful talk, and I very much enjoyed the interaction between the genes, and especially I'm kind of interested in the ARMS2 H1 locus, and I was thinking, first of all, whether you consider that ARMS2 can be a long non-coding RNA or short non-coding RNA, and how it could have a work. And my second part of the question is whether you were thinking to look at the whole locus. So it's like a whole domain which has only 3 genes. Well, actually, ARMS2, H1, and some other non-coding RNA, and whether to maybe look how this changes in the disease RPE versus healthy RPE, whether there is an interaction between those 3 open reading frames.
You mean an interaction between the genes that are found within that region? Is that what your question is?
Yes, and I actually mean both interaction on the level of the chromatin and also on the genetic level.
Okay, so I'm going to tell you something that's really interesting. That is something that we just observed, and I don't know what to make of it. When we looked at the ARMS2 A69S animals, we saw this upregulation in the RNA for HTRA1. But we also saw an upregulation of P10, which is the other gene that has been recombined out of the area. So it's almost as if, so I talked to somebody who's a specialist in transcription and looking at genes, and he says sometimes when you introduce a gene to an area, you can affect across the region by that insertion, the entire region, genes within that entire region. So what we really need to do is to take the ARMS A69 animals and ask whether or not they also show an increase in HTRA1 and in the P10, right? So is it that we've just introduced something and that's changed something in basically like in the entire region, or is it specific to this particular allele? And that's something that we do need to look at still. But your point is well taken. It is possible that there's something that is happening at very basic level in these animals.
Sam?
Yeah.
Thank you for your wonderful talk. My question is, have you looked to see if rods and cones are differentially affected in your mutant mice?
If rods and cones are,
Differentially affected by staining or ERG?
we have not gone that far, but that is definitely something that we ought to do in relationship, especially to the ARMS2 gene. But it's like, I think about this a lot, right? When you work with a model system, it's just that, a model system, right? It's not to say that it's going to be identical to what is observed in humans, and there are likely to be phenotypic characteristics that occur in humans that you don't see in mice, right? But it doesn't make it less of interest in studying the disease of these animals. As I mentioned, you can look at subphenotypes. So yes, we should look at the differential effect on cones and rods, but we have not done that. And it could very well be that it might not have an effect. And I think the other point to be made is that these are model systems and they're not going to totally recapitulate the disease that is observed in humans. And when I think about this, I thought at first I was like, oh dear, here we have this model. And then it occurred to me that in humans, there's a mixture of genes that are causing disease, right? It's the interaction of multiple variants. So it's not surprising that in an animal model of a single variant, why would you expect to see the entire phenotype? It's sort of going off script here, but I just want us to think a little bit about the fact that these are model systems and not all phenotypes that are observed in humans that are genetically heterogeneous are going to recapitulate in these models. And my husband, Jürgen Nagert, would argue that it's because you didn't put them in the right genetic context, right? And so you may not see something in Black 6, but perhaps if you cross it to NOD or to DBA, you might see something. Anyway, that wasn't necessarily a question, but I just thought I would interject that.
John?
Hello, I was wondering if, ARMS2 being a mitochondrial protein and it being one of the very handful of genes mitochondrial genes in the human genome, I was wondering if, depending on the mutation, it leads to issues with proper protein expression and also localization to the mitochondria itself. And then also my second question is if you look into just on the wall type arms to protein-protein interactions, like complexes with the maybe in mitochondrial isolate.
Okay, so to your first question, I think it's actually quite controversial, the localization of ARMS2. Some groups have shown that it might be in the mitochondria, and other groups have suggested that it's not. There are no good antibodies currently that are available, and that's one of the reasons that we decided to work with Dr. Ufing, because he has an antibody, and we wanted to know in our model system whether or not we'd be able to localize ARMS2 to the mitochondria. We don't know whether or not it's localized to the mitochondria, and in fact, there have been groups that suggest that it's in monocytes. And in the capillary pillars, secreted to the capillary pillars of the choroid. So it's not a given that it is localized to the mitochondria. That's the first thing. The second thing is, now that we have, these two model systems, it will make it possible for people that have expertise to look to see whether or not there are actual biochemical interactions between the variant versus the wild type allele. That's not something that is our expertise, but that's one of the reasons that we make our models available, to the world.
Okay, let's go to Zoom. Mike?
Yes, thank you. Just really fascinating that the area centralis may be in the dorsal retina, and I'm just wondering, what other work have you done or anticipate doing to support this conclusion?
Well, if we had all the hands in the world, I probably would try to look structurally in the retina to see whether or not there are differences in expression in those cells that are in the dorsal region versus other areas of the eye. That would be one thing that could be done. We have not done that, but we have talked about doing that. There's something different, obviously, about these cells, or the structure in which they sit, that cause them to develop disease. Because when you think about it, all cells should have this mutant form of TBRM5, of ctNNA, and yet it's only this region that appears to be susceptible. So, I guess it's sort of like, people have done, like, for example, in macular disease, they isolate the macula and then they look at the periphery and look for differences. And those experiments can be done. It's not something that is like at the top of our priority list at this time, but it is something that can be done. And I don't know if you have other suggestions of how we can try to determine whether this is the case.
Okay, let's move to Jean.
Hello, okay. That's fantastic talk. Thank you so much. I have a question about your HTRA1 stuff. So compared to the black 6, the level of HTRA1 in the RPE of your ARMS2 A69S mice are increased. So my first question, in your mind, this level of this HTRA1 is upregulated by your insert genes or not, or because in the 2021 PS paper from the Gregory of Utah, they suggested that reduced the expression of HTRA1, mRNA and the protein in the retina RPE of chromosome 10 risk donor was be found, and also related with the AMD. So how do you think about the relationship between your mouse model and this human data? That's my questions.
Yeah, so I would say that, you know, there are some people that have shown downregulation of HTRA1, and some people have seen upregulation of HTRA1, and some people have seen no effect on HTRA1 in face of having the A69S allele there. I don't know, Anand, do you, I mean, what do you think? I think it's pretty controversial, right, at this time.
Yeah, Chris asked me to give the last comment, so I'll wait till everything else is done, right?
Okay.
Brother.
Yeah, I mean, I think the thing is, right, the experiments that we're going to do, okay, so as I mentioned earlier, you know, there are other genes that are upregulated in that area. And so it could be something that is very, very basic to the expression and transgenic, well, to the insertion of the transgene into the region. And that may be causing the elevation HTRA1 in these models, but we'll be able to formally test that by looking at the A69S, which we haven't done yet. And I think when we do the experiments to knock down HTRA1 in the presence of A69S, that might give us some information as to whether or not A69S is acting through misregulation of HTRA1. But I think, yeah, I mean, this is data as it is. You know, I always tell my people that data doesn't lie, right? It could be that, yeah, so this is what we see. Other people may see nothing, and other people may see, as Greg has, a decrease. So it's hard to be able to say, but this is our data as it is. Right?
Great. So the last question is, does that HTRA1 level in the retina also be increased or decreased, or something doesn't change?
I feel like saying it did not,
Okay.
but I would have to go back and look at the data. I'll write your name down so that I can get back to you.
Yeah, my name is Ying. Is from Elegant.
Excuse me?
My name is Ying, Y-I-N-G, last name Y-U.
Hmm? And where are you at?
Yeah, I'm from the Elegant, the AbbVie, the company.
Okay.
Thank you.
Maybe you can email. Maybe you
Yeah,
can email.
I will
me.
email you. Yeah, I will email.
And then I can try to give you that information. Right.
Thank you so much. Appreciate it. Thanks.
Arjuna.
Thank you. Thank you for sharing that. It's a really interesting talk. I just wondered what your take on is with regards to the impact of age on these models. So obviously, you know, you've got a strong component of aging in most of these retinopathies, but mice being what they are, you presumably age them up to, you know, 2 years on best. Where do you think that plays a role in this? Thanks.
I mean, as I mentioned, you know, like when you look at the black 6 animals for the fundus spotting phenotype, it's there at 18 months of age. And it's just more severe in the presence of A69S. I'd also point out that it's not fully penetrant. The phenotypes that we observe are not necessarily fully penetrant. And I don't really know why that is, whether or not it could be, yeah, I don't know why that is, but it's not fully penetrant, disease phenotypes that we observe. But I think aging is definitely a component of the disease. And yes, it's a problem that mice only live to 2 to 3 years of age. But I think that we can get an inkling that there is disease progression by looking across time. Yeah. But I agree, that is a problem.
Jensen.
Yes, hi, Patsy. My question, I have a question about your alpha-ketinin mutants. I'm wondering if you have looked at the canonical Wnt activation in the RPE, since alpha-ketinin normally restrain that pathway. And if it happens, that could stimulate epithelial to mesenchyme transition. The RPE would migrate and piling up and all of that. And that is consistent with the picture that you shown. And I was just wondering if you have looked into that as a plausible pathogenic process.
I feel as if we did at the very onset when we looked at the mutant. And I feel like there wasn't a difference because that was an obvious possibility. And I don't think we saw an effect, at least in the Wnt markers that we looked at by transcriptomics. But I can definitely get back to you and confirm whether or not that's true. So this is Jason.
This is Tiansen Li, Patsy. Yeah.
Tiansen Li, hi.
Hi. Yeah. Okay. Great. Thank you for answering my question.
I will. So this is Wnt, whether or not we saw change in regulation.
So we have final 3 questions from the room, and then we're going to highly anticipated final comments from my brother Anand. So let's go to Philip.
Hi, Dr. Nishina. Really nice talk. I really enjoyed it. Actually, Mike took my question, so I'll just compliment you on the work you were doing with the TBMR5, and I think it's really impressive what you're doing with that huge number of crosses. It kind of blew my mind whenever you said that. So good luck with those experiments.
Thank you.
All right, Emily.
Hi, Dr. Nishina. Thank you for a great talk. I was just curious, and maybe I missed this. In your ARMS2 mutant, I was just wondering if you saw any sex differences in any of the subphenotype progressions.
We always look at males and females, and we haven't seen an effect either with ARMS2 or CTNNA1, but we do have models in which we do see sex differences. But there's nothing actually overt that stands out in my mind about either of these models in relationship to sex differences.
Regina.
Hi, Dr. Nishina, beautiful talk. And I was intrigued by the images of the RPE in your mutants, which show large number of multi-nuclei RPE cells with more than 2 nuclei. Is this also the case in humans?
I think in human donor cells, they have shown, and you need to correct me, Anand, if this is not true, that there is areas of multinucleation. This is one of the phenotypes that have been observed in AMD donor eyes.
And now, ladies and gentlemen, let's move to Dr. Swaro.
Drum roll!
Hi. Yeah, hi, Patsy. First, wonderful as always. I think you are doing some of the most beautiful work using mouse genetics. I mean, that's at the best with all these multiple mutant. I'm sort of course, going to comment about ARMS2 HTRA, which is sort of something I still feel is completely unclear, as you said clearly, but your work is sort of in the right direction. But first, let me comment on something that Dorota was saying, and you said, that is definitely true, that genome architecture, the topology in the region will be altered if you have an insertion or a deletion of any kind. So it could be, particularly if you have a large enough deletion, it could change that. But really, I think one of the things that one can do is look at the high C or kind of architecture, which use one of the high C technologies or some, or maybe more directed 3 or 4 C kind of technology to look at the region where you have and show that that is not really having any drastic change. And if you could do that, then you can argue that whatever you're seeing is because of what you have added and not because of alterations in chromosomal architecture. So I think some of those things require a little bit of work, but that sort of will clarify that. But you know, your idea of putting both arms to and the mutant wild type in this is very clever. And I have to tell you that we thought of that, but didn't dare to do it because I thought it would take a lot of time and effort, and I don't know what we will get. Arms to, we had made 10 plus different antibodies, and only 1 or 2 antibodies worked, and we actually had localization in mitochondria with those antibodies, and these were using human tissues. We did some work with Bob Ferris, who Chris knows very well, and one of my postdocs, and we sent those antibodies to a bunch of people, and couple of people could repeat it, some others could not repeat it. And in the meantime, those antibodies, polyclonal antibodies from rabbit got over, and we sort of gave up. We were doing some in vitro experiments where we did tons of deletions, actually probably 10, 15 deletions from N-terminal, C-terminal, in between, and all that in the protein. And we actually could show that the protein does localize to mitochondria, at least in cultured cells. And you know, if you remove this N-terminal mitochondrial sort of signal, then it goes away. So we had done huge amount of work, but we didn't publish. This was done some 10 years ago because we could not show that in vivo it's in mitochondria. So that controversy remains. And since the expression of ARM2 is so low, at least in these, you know, at the protein level, it's so hard to really clarify. So all the work that other people are saying about HTRA, I think is valid, but that doesn't mean the controversy is resolved in any way. It could be both genes. It doesn't matter. I mean, you know, at one locus, there could be 2 genes contributing to AMD, either independently or by some sort of interaction. Genetic mapping does not preclude that only one of these could be, or exclude that only one of these is responsible. So I think, you know, there were 2 reviews recently in progress in retinal eye research, one on HTRA from Iwata and other on ARMS2 from Maria Sufing, who had a different take on ARMS2. So I think there's just a lot of work going on, and there's no consistency. Unfortunately, different labs have found different things. We never found in human RNA studies any change in AMD patients, either in ARMS2 or HTRA. We did some bunch of retina 40, 50, and we didn't see any change. So I don't know what to say. So I think what you are doing is exactly what we need to do, use model systems to understand the biology, and maybe both ARMS2 and HTRA contribute to AMD either independently or in some sort of interacting manner. I don't know. I mean, I'm not going to resolve that. We spent huge amount of time, and then sort of, I think we have to do at the protein level somehow. We had to find some good antibodies, and multiple groups have to show what it is.
Right.
Yeah. So I'll just stop there. It's just sort of, but it's a wonderful way you are doing, and I think your work always sort of is admirable. So thank you.
And with that, again, Patsy, thank you so much. It was wonderful, wonderful presentation and work. Thank you, Alan.